Good morning, Vananda. Myself, Uma Maheshwari Kolipaka. Today, I am going to discuss about PCR, polymerase chain reaction, which is very important topic in biotechnology, pharmaceutical biotechnology. That is a part of B Pharmacy third and sixth semester. So, first coming to contents, what is PCR? Today, we are going to discuss all these topics related to PCR. First of all, what is PCR? History of PCR, components of PCR, principles of PCR, basic requirements, instrumentation, PCR program, advantages and applications of PCR. So first coming to the understanding of PCR. PCR is a technique that takes specific sequence of DNA, that takes a specific, that is our desired sequence of DNA we are taking. That is a small amount, a small fragment of DNA we are taking, that we are amplifying with the help of this PCR, polymerase chain reaction. So we can define PCR. It is a technique that takes specific sequence of DNA, a small amount or small fragment, and it can be amplified, which is used for the further testing in vitro technique. In vitro techniques, that is PCR is in vitro technique, and with the help of PCR, we are amplifying the small fragment of our desired gene into n number of copies so that we can come to know various details of that sequence of that desired fragment. And sometimes we can come forward by identifying the genes exactly in our desired way. That is all we are doing in. So this is specifically in vitro technique. Next, coming to the short history of PCR. PCR is developed in 1983 by Dr. Carey Mullis. And first publication of PCR by Cetus Corporation appears in Science. And purified tech polymerase is first used in PCR in the year of 1986. So here we need to understand what is tech polymerase. Tech polymerase is nothing but an enzyme which we are using in polymerase chain reaction. That is, we are extracting from the bacteria Thermus aquaticus. So tech polymerase is purified first time in 1986. And Perkin-Elmer introduces the automated thermal cycler. Thermal cycler is a machine which we are using in the technique of PCR. Science declares tech polymerase molecule of the year in 1989. So basically, PCR is also known as molecular photocopying. Generally, if you are observing general photocopying, that is Xerox machines, what they can do, they can just make the number of copies of already existed one. So same in the same manner, in the same way, if we are having our desired gene fragment, we can amplify, we can make n number of copies of that fragment with the help of this PCR. That is the actual meaning behind the PCR, polymerase chain reaction. So polymerase chain reaction is first and foremost developed by Dr. Carey Mullis in 1983, and Cetus Corporation appears in Science. That is first publication of PCR by Cetus Corporation. Publication when the PCR published first time, the information regarding PCR was published by Cetus Corporation, that is appears in Science in the year of 1985. Next, tech polymerase is first time used and purified in PCR. So first of all, tech polymerase is an enzyme that is extracting from a specialized bacteria, Thermus aquaticus, and this enzyme is purified for the first time, that is used in PCR in the year of 1986. Next, Perkin-Elmer introduces the automated thermal cycler machine. This is also known as DNA cycling machine or thermal cycling machine or PCR machine. which is actually introduced in the year of 1988. And science declares tech polymerase, that is molecule of the year. Tech polymerase is having a lot of importance in PCR. To be frank, without tech polymerase, we can't complete the process. We can't do the PCR technique. So this is said to be molecule of the year in the 1989. Next, amplification and detection of specific DNA sequences using a fluorescent DNA binding dye, laying the foundation for future real-time or kinetic PCR. That is happened in the year 1990. Amplification is nothing but making n number of copies, multiplying the copies, and detection of specific DNA sequences using a fluorescent DNA binding dye, a fluorescent DNA binding dye, laying the foundation for future real-time or kinetic PCR. Generally, PCR is said to be or categorized into different types: real-time PCR, RT-PCR, kinetic PCR, like this. So, in that way, in this real-time or kinetic PCR, we are using the fluorescent DNA binding dye. So, that is happened in the year 1990. That is used for the amplification and detection of specific DNA sequences. And in 1991, in the year of 1991, RT-PCR is developed using a single thermostable polymerase, RTTH, facilitating diagnostic tests for RNA viruses. So, in the year of 1991, what happened? RT-PCR, reverse transcriptase polymerase chain reaction, is developed using a single thermostable polymerase. Thermostable means it can able to tolerate the changes in the temperature. Thermostable polymerase, RTTH, facilitating the diagnostic test for the RNA viruses. Not only DNA, RNA viruses also diagnosed, facilitating the diagnostic test for the RNA viruses. In the year of 1993, Dr. Carey Mullis shares Nobel Prize in chemistry for the conceiving PCR technology. So, in the honor of the Carey Mullis, Dr. Carey Mullis, he honored with the Nobel Prize in the year of 1993 for his inventions in respect to the Department of Biotechnology. That is, by introducing the PCR technology, he honored with the Nobel Prize. Next, Dynial launches DRB36 HLA typing kit for the tissue typing. So these are all the biotechnology process by using which we are using to detect the different types of genes, different types of tissues, and detection of different diseases, gene sequences, etc. So HIV-1 monitor test version 1.5 product family that is happened in the year of 2003. And Amplicor CT-NG test for Chlamydia trachomatis, Amplicor CT-NG test for Neisseria gonorrhea. These are all happened. That is as a part of history of PCR. Next, coming to the principle of PCR. So when we are going to history of PCR, basically we need to remember the scientists, at least Dr. Carey Mullis, who developed the PCR, that is happened in the year of 1983, and the tech polymerase enzyme that is purified first time in the year of 1986 and used in PCR technique. And this is said to be molecule of the year in the year of 1989. So these are the things we need to at least remember in respect to the history of PCR. Next, coming to the short history of PCR, that is HLA typing and Chlamydia trachomatis amplicor, Neisseria gonorrhea amplicor, and HIV-1 monitor test. These are the years of developments. Next, principle of PCR, purpose. What is the purpose of PCR? What are the different conditions we need to maintain? And what are the different components we need to remember in respect to the PCR? We are going to discuss. First, coming to the purpose. To amplify a lot of double-stranded DNA molecules with the same size and sequence by enzymatic method and cycling condition. First of all, what is the purpose of PCR? If you raise a question, that is, what is the purpose of PCR? Why we are doing this PCR? Why we are going to polymerize the chain reaction, why we are going to amplify, why we are going to multiply the number of copies, that is with the help of already existed one. Means the answer is to amplify a number of double-stranded DNA molecules, that is fragments with the same or identical size and sequence by enzymatic method and cycling conditions. Sequence and by same size and same, that is similar sequences in respect to the size and shape or length and sequence by enzymatic method and cycling condition. So we are making n number of cycles, we are completing n number of, as we are completing the number of cycles, we can get that number of increased number of copies that are double-stranded DNA molecules already existed one. If you consider a small fragment of gene, a small fragment of nucleotide sequence, if you are having, and if you wanted to multiply that small fragment into n number of copies, you have to use this PCR technique. So we can conclude the purpose of PCR is just to amplify, just to multiply the number of copies, just to multiply a small double-stranded DNA fragment into n number of copies. That is the basic purpose of PCR. That is, we are doing with the help of cycling conditions and by using various enzymes. What are the different steps involved in the PCR technique? So first of all, denaturation. Basically, 3 steps we have to come across during PCR technique to get n number of copies of DNA. So first step is denaturation of double-stranded DNA template. Denaturation of double-stranded DNA template. That is the first step. And second step is annealing of primers. Annealing, streaking or attaching of primers. Next, third step is extension of double-stranded DNA molecules. So these are the 3 steps. Denaturation. Briefly, if we wanted to ask me to say briefly the steps involved in the PCR, simple steps, that is denaturation, annealing and extension. First, coming to the denaturation. Denaturation means separation of 2 strands. Basically, we are taking double-stranded DNA fragment. Our desired DNA fragment we are taking, that is DNA, double-stranded DNA we are taking. So this double-stranded DNA, with the help of denaturation, we are separating the 2 strands, the complementary strands, which will fold it 1 to 1, that is helical structure we have. So that 2 strands we are separating from each other. That process is known as denaturation. So basically denaturation will be taking place at the temperature of 92 to 94 degrees Celsius, and double-stranded DNA melts to the single-stranded DNA. This is denaturation. Next, coming to the annealing process. Annealing temperature, that is 50 to 70 degrees Celsius temperature we need to fix to anneal the primers. Here we are taking the primers. These primer sequence will come and attached or annealed here to the already existed one. Here also, this is the primer. This primer will get binding, will get attached, will get annealed to this already existing single-stranded DNA molecule. This process is known as annealing, and this will be taking place at the temperature of 50 to 70 degrees Celsius. Next, coming to the extension. Extension is nothing but this is also happening at the temperature of 72 degrees Celsius. Already primer existed in the annealing step. To this primer sequence, we are taking the different types of nucleotides and tech polymerase enzyme. So with the help of tech polymerase enzyme, these nucleotide sequences will come and bind here, that is as a sequence of the primer, so that the complete strand will be formed. So this requires the time of 0.5 to 3 minutes, and DNA polymerase bind to the annealed primers and extends DNA at the 3' end of the chain. So basically, we know 3' to 5' end, 5' to 3' end, these two are complementary to each other. These are all very basics. I hope you are aware about this. And when coming to the annealing process and extension process, in annealing, the primers are get attached to the already existed strands in a reverse manner, that is 3' to 5' end. If the primer is get attached to the 3' end, that is opposite primers will come and bind to the existed strands. 5' primer will come and bind at the 3' end, and 3' end of the primer will come and bind at the uh, 5' end. So in that way, primers already get attached to the existed strands in annealing process. When coming to the extension, with the help of tech polymerase enzyme, different types of nucleotides, according to the sequence which is already existed on the single strands, according to the sequence, exactly complementary nucleotides will come and bind to that existed strand with the help of tech polymerase enzyme at the temperature of 72 degrees Celsius. This is actually extension. This requires the time period of 0.5 to 3 minutes. Next, this is a cycling machine. First of all, we are maintaining the temperature in thermal cycler machine, that is 90 to 95 degrees Celsius. If you want specific temperature, that is about around 92 degrees Celsius. So that first of all, we are maintaining the temperature 92 for the process of denaturation of existed double-stranded DNA to get separated into the single strands of DNA. This is 92 degrees Celsius, at which we can expect denaturation. Next, we are decreasing the temperature. That is, we have already explained, that is, we are already come across with the temperature for annealing process, that is 50 to 70 degrees Celsius. So, we are decreasing from 92 to 50 to 70 degrees Celsius. This is around 52 to 54 degrees Celsius, like that. Then again, at this temperature, 50 to 70 degrees Celsius temperature, annealing process will be taking place. That is, primers will come and attach it to the opposite directions to the already existed strands, single strands. Next, we are again increasing the temperature to the 72 degrees Celsius. So at this temperature, the extension process will be starts and complete to form the complete strands. That is, single stranded DNA will become double stranded by forming the new strand. Again, we are increasing the temperature. So by this extension, we can understand here the one cycle of PCR has been completed. Again, when we are increasing the temperature, again this will denaturate the existed double-stranded DNA molecules. This is the actual plot we can, this is the plot we can draw by taking time on x-axis and temperature on y-axis in respect to the PCR procedure. So products of extension, basically, first of all, we have taken requirements, that is tech polymerase enzyme, nucleotides, primers, and already existed DNA desired DNA fragment or DNA desired small genes, small portion of the double-stranded DNA. These are all the things, a small amount of buffer solution we are taking in thermal cycling machine. So we are maintaining the different temperature conditions so that we can expect the three steps, that is denaturation, annealing, and extinction. So that first, by the completion of one cycle, two Initially, we are taking only one double-stranded DNA fragment that is getting separated and forming new strands, so that by the completion of the one cycle, we are expecting the two double-stranded DNA molecules. So within very short span of time, we can expect the n number of copies by repeating the cycles. So already existed strands will get separate, and with the help of primers, nucleotides, and tech polymerase enzyme, n number of copies are forming. Again, the cycle will complete. Again, we are increasing the temperature to 92 degrees Celsius, so that again these existed strands will get separated into single strands. Again, the cycle will starts and uh, helps to form the n number of copies. So, what are the chemical components we require for the process of PCR? Magnesium chloride 5 to 2.5 millimeters in buffer, that is to maintaining the pH 8.3 to 8.8, that is slight alkaline pH we require. And dinucleotides, that is dinucleotide phosphates 20 to 200 micrometers length, and primers 0.1 to 0.5 micrometers length. And DNA polymerase that is 1 to 2.5 units. Target DNA that is less than 1 micrograms. These are the components we need to mm, these are the prerequisitions for the starting of PCR technique. Basically, we have to incorporate all these things into the tubes, that is we can put into the thermal cycling machine, so that that's next. Basic requirements for the PCR reaction: DNA sequence of target region must be known. So if you wanted to detect, if you wanted to do research in respect to the any disease or any sequence, that is for the identification of parenthood or for the identification of culprit in a crime scene, whatever it may be your requirement, first of all, you have to identify, you have to decide what is your desired fragment, what is your desired sequence of nucleotides that is said to be the DNA sequence of target region. So we must know the target region. Next, primers, typically 20 to 30 bases in size. These can be readily produced by commercial companies, can also be prepared using a DNA synthesizer. So when coming to the primers, these are commercially available in the market, so we can procure them. Otherwise, we can synthesize them with the help of DNA synthesizer. Next, thermostable DNA polymerase. Actually, here we need to discuss about the tech polymerase enzyme. Tech polymerase is the enzyme that is already, I told you, this is we are extracting from the Thermus aquaticus bacteria. And what is the specialty? Why we are only depending on the tech polymerase enzyme? Because tech polymerase enzyme can able to tolerate high temperature. Basically, whenever we are talking about the enzymes are optimum temperature for the maximum catalytic activity of the enzymes is said to be 40 to 45 degrees Celsius, or in some textbooks 35 to 45 degrees Celsius. So I hope you are aware about the optimum temperature. Optimum temperature means that is the suitable temperature for that enzyme to exhibit its maximum catalytic activity. So in respect to that, most of the enzymes will be having the optimum temperature around 35 to 45 degrees Celsius, even though some exceptions are there. So according to that, our tech polymerase enzyme is exceptional. This tech polymerase enzyme is specialized in such a way that can able to tolerate the higher temperatures, 90 to 95 degrees Celsius. So actually we need this high temperature for the purpose of denaturation, that is to separate the double stranded DNA into two single strands. So for this purpose, we are using the tech polymerase enzyme. Otherwise, if you are using any other enzymes that only having optimum temperature around 45, 35 to 45. So when we are increasing the temperature, the enzyme will become inactive, or the catalytic activity will be reduced in a drastic way, so that our intention will not be achieved. So DNA thermal cycler machine, which can be programmed to carry out heating and cooling of samples over a number of cycles. So thermal cycling machine is also important, which can be programmed to carry out the heating and cooling. We are fluctuating the temperature. We are changing the temperature within very short span of time. So according to these fluctuations in the temperature, actually our process, that is denaturation, annealing or extinction, will be taking place. So in such a way, we are designing the thermal cycler machine. So this is very, very important. Without thermal cycler, we can't make number of copies of DNA. Here you can observe the machine. This is the instrument actually we need. A thermal cycler machine, or a PCR machine, or a DNA synthesizer. Like this, various names are there for this machine. So we can just taking the things, and we can incorporate the things, all requirements, buffer solution, take polymerase enzyme, and piece primers, and already existed DNA strand, our desired or targeted gene fragment into the tube, thermal cycler tube, and this tube we are placing into the machine, and we are maintaining the temperatures. We are fluctuating or changing the temperature from 92 to 50 to 70, 50 to 70 to 72, like that. We are changing the temperature so that our desired process, that is denaturation, annealing, and extinction, will be taking place. So that we can observe the number of copies, that how they are forming within very short span of time. So three aspects of PCR, that is specificity, efficiency, and fidelity. Actually, specificity means a specific DNA or specific target fragment we are taking. The same sequence we are multiplying, the same sequence we are making into a number of copies. Next, efficiency. Efficiency means within very short span of time, in a efficient manner, in a very efficient way, we are making a number of copies. That is nothing but efficiency. Next, fidelity. There is no mismatchings or any wrong sequences we are making. The same sequence, that is very much identical in size or in nucleotide basis, and the same things we are making the identical copies into n number. These are the three aspects of PCR. Next, things to try if PCR does not work. What what are the things we have to try if PCR is not working? If no product produced of correct size, check DNA quality. First of all, we have to check the DNA quality, whether we have taken the quality DNA or not. Reduce annealing temperature. So we have to reduce the annealing temperature will be having a wide range, that is 50 to 70 degrees Celsius. So we have to adjust the annealing temperature and increase magnesium concentration. Add dimethyl sulfoxide to SA at around 10% and use different thermostable enzymes. Throw out primers, make new stocks. If we are feeling that the production of n number of copies is not as per our requirement, in at that situation we have to change the primers. Already we have taken some sort of primers, but if we are feeling that these primers are not working, so in that case we have to change the primers. Next, if extra spurious product bands present, increase annealing temperature, reduce magnesium concentration, reduce number of cycles, and try different engine. So these are the things we have to try if we are feeling that PCR is not working. An example of PCR program. What is the exact time required for the different steps? Here we can observe initial denaturation 95 degrees Celsius for 5 minutes. 95 degrees Celsius for 5 minutes, and thermocycle file 30 cycles, and denaturation 95 degrees Celsius for 30 seconds, annealing 55 degrees Celsius for 30 seconds, extension also taking only 45 seconds, that is at the temperature of about 72 degrees Celsius, and final extension that is taking 5 minutes at the temperature of 72 degrees Celsius. So holding soak file usually 4 cycles. So initial denaturation taking 5 minutes, thermocycle file 30 cycles, uh, next denaturation 30 seconds, annealing 30 seconds, extension 45 seconds, final extension 5 minutes. So these are all matter of minutes or seconds. Especially denaturation will be very short span 30 seconds, annealing 30 seconds, and extension 45 seconds. For some time, a final extension and initial denaturation, we are taking 5 minutes time. Advantages of PCR: small amount of DNA is required per test. Result obtained more quickly, usually within 1 day per PCR. Usually not necessary to use radioactive material, that is phosphorus 32 for PCR. PCR is much more precise in determining the sizes of alleles essential for some disorders. And PCR can be used to detect point mutations. So these are all the advantages. Small amount of DNA is required, so we can easily take the small amount. We are not depending on the much quantity of DNA, and result obtained more quickly within very short span of time, within 1 day, not actually 1 day or 1 hour, within minutes we can get the number of copies. So usually not necessary to use radioactive materials like radioimmunoassay. We are not taking any sort of radioactive materials here. And PCR is much more precise in determining the sizes of alleles and essential for some disorders. So PCR can be used to detect point mutations. And applications of PCR: Neisseria gonorrhea, Chlamydia trachomatis, and HIV-1, factor V Leiden, in forensic testing and many others. So we can detect different types of various genetic disorders, parenthood identification, and culprit identification in crime scene. Number of applications are there. Apart from that, forensic testing and factor B Leiden and HIV-1, chlamydia trachomatis, Neisseria gonorrhea. These are all the applications. At least we need to take down the notes. And applications of PCR: molecular identification, sequencing, genetic engineering. Archaeology, epidemiology, ecology, fingerprinting, DNA fingerprinting, classification of organisms, genotyping, prenatal diagnosis, mutation screening, drug discovery, genetic matching, detection of pathogens, and sequencing, bioinformatics, genomic cloning, human genome project, and genetic engineering, site-directed mutagenesis, and gene expression studies. So these are all the areas we can utilize PCR in very specialized way. So PCR is a wonderful technique which we are using in the Department of Biotechnology for these various applications. So last, coming to the conclusion, PCR is not only vital in the clinical laboratory by amplifying small amount of DNA for sexually transmitted diseases detection, but it also important for genetic predisposing for detects defects such as factor 5 Leiden. The PCR technology can also be employed in law enforcement, genetic testing of animal stocks and vegetable hybrids, and drug screening, along with many more areas. So this is the all over this year. Thank you.